Hi everybody. In this unit we'll be talking about the other excitable tissue inside the body, which is muscle, or more specifically about skeletal muscle. As you'll hopefully see, skeletal muscle follows a lot of the same patterns that we've already introduced back in our neurophysiology unit. I'm going to start by introducing skeletal muscle as a tissue, and then I'll move into talking about some of the microanatomy and about the molecular basis of contraction, the sliding filament model and excitation-contraction coupling. Towards the end of the unit, we'll zoom back out to talk briefly about muscles and gross anatomy and a little bit about muscle mechanics. So the general properties of muscle are excitability, which fortunately we've already talked quite a bit about. Muscle generates graded and action potentials through the same mechanisms as neurons. But unlike neurons, which use their changes in membrane potential to release neurotransmitter, muscles use their action potential to initiate the process of contraction, which generates force. Muscles are constantly changing size due to their elastic properties. So muscle does have a resting length, but unlike most cells inside the body, it doesn't lose its membrane integrity when it experiences large changes in size. And even though most often we think of these changes in size as shortenings of the cell, muscle is also an extensible tissue, so it can be safely stretched beyond its resting length as well. Now, most of the descriptions of muscle that we've given you up to this point have dealt with its major function, which is movement. Muscles are attached to bones, and so therefore when muscles contract, they can create all sorts of interesting motions that I'm sure you've been spending the past couple of weeks trying really hard to memorize. But we also have muscles that are contracted to some degree, at least all the time. And those muscles are particularly important for creating posture, supporting our posture inside the body. And lastly, because muscles are doing work, when they contract, they produce heat as a byproduct. And that makes muscle really important for thermoregulation, or maintaining temperature homeostasis inside the body. And that's the reason why you shiver when you're cold. So, like neurons, muscle is a terminally differentiated tissue. And that means that muscle doesn't go through mitosis. But muscles still need to grow, and muscles get damaged, need to be repaired. So we have to have a mechanism to generate new muscle cells inside the body. And that's what we're taking a look at on this diagram here. So creating a muscle starts with the differentiation of a multipotent stem cell within muscle tissue called a myosatellite cell. These stem cells eventually become an immature muscle precursor that's known as the myoblast. That's the cell that's up at the top of this diagram. And if you look closely, you can see that morphologically, myoblasts resemble some of the other cell types that we've talked about back in the tissues unit. They don't look particularly muscly. But as this maturation process continues, these tiny myoblasts line up next to each other end to end, and they form a tube-like structure that's ultimately going to become a mature muscle cell. Mature muscle cells are also known as myocytes and are also known as muscle fibers. In fact, one of the more confusing things about muscle is that people tend to use these three terms interchangeably because they all refer to the exact same thing. On this slide here, we're comparing the three major types of muscle inside the body: skeletal muscle, smooth muscle, and cardiac muscle. Now, we're going to talk a lot more about smooth muscle and cardiac muscle in the next half of the course. So the focus of this unit is really on skeletal muscle. And the only thing we'll need to be able to do with those other two types of muscle is differentiate them under a microscope from skeletal muscle. So if we were thinking about that, we could say, for example, that skeletal muscle is the only multinucleate tissue, meaning that it has 3 or more nuclei per cell. And it actually kind of makes sense if you think about that developmental pathway that we just talked about a second ago. Each of those individual myoblasts has its own nucleus before it fuses to form the muscle fiber. So therefore, the muscle fiber, made up of many individual myoblasts, has to have a lot of nuclei. You can also see that both skeletal muscle and cardiac muscle have an overall rectangular or cylindrical type shape to them. They also have vertical lines known as striations going down the short axis of the cell that we can see under a microscope, and we'll be talking a little bit more about those later. Lastly, you can see that skeletal muscle is a pretty cellular or densely packed tissue. There's very little space between two adjacent skeletal muscle cells. Now, we're going to spend most of our time in this unit thinking about muscle from a cell biology perspective. And that's going to make it a little bit different than what you've just learned in lecture and what you've been covering in the lab. To try to bridge this gap, this slide here is showing you that muscles are organized at multiple levels. Similar to a compact bone, skeletal muscle is really a system of tubes within a tube. So a muscle in gross anatomy, like for example your biceps, is made up of a bunch of tube-like structures called fascicles. Each fascicle is a bundle of muscle fibers that's held together by a connective tissue wrapper. And even at the subcellular level, each individual muscle fiber contains myofibrils, which are created from the arrangement of the contractile proteins inside the cell. And again, we'll talk more about those a little bit later on. Now, there can be some variability among muscle fibers, even within a single muscle. So as one example of this, we could think about differences in metabolic properties of muscle fibers. For those of you that are carnivores, if you look closely at your steak next time you're in the meat department at the supermarket, you'll notice that even though it has an overall reddish appearance to it, if you look closely, you can see that the individual muscle fibers, which you'll be looking at in cross-section, are actually a mosaic. And if you put this thin cross-section under a microscope, which is what's happening on the left-hand side of the slide, you can see that the red appearance overall is actually created because some of the fibers are dark red, some of them are more pinkish, and some of them are actually white or translucent. And the difference in color is actually because of an oxygen-binding protein called myoglobin. If you've heard of the protein hemoglobin before, myoglobin is its counterpart inside of skeletal muscle. Now, the fibers that are the darkest red are known as type 1 fibers, and they have the most myoglobin. And that means that they're really good at binding and trapping oxygen in the tissue. And that allows them to produce a lot of ATP, and therefore they can stay contracted for a long period of time. Postural muscles and muscles that are used in endurance activities are typically made with a lot of these slow muscle fibers, these type 1 fibers. At the other extreme, the more translucent type 2b fibers have little, if any, myoglobin. And that means they rely entirely on anaerobic respiration, which is a lot less efficient at producing ATP. So this means that these fast fibers can only be used for short bursts of activity like sprinting or weightlifting. The type 2a fibers, which have some myoglobin, are considered to be intermediate. They have the ability to do a limited amount of aerobic respiration.